Hi, my name is Joan, and this is my project on the effects of varying light quality on leaf Nyctinasty on East Acacia. So what is leaf Nyctinasty? Also known as sleep movement, leaf Nyctinasty is the closing of leaves following the plant's internal circadian rhythm, which is influenced by the changes in sunlight received during day-to-night cycles. While there is some research about leaf Nyctinasty, not much known is about why the plant evolved to have this trait and the plasticity of it under different light quality conditions. Through this study, scientists can have a better understanding of Senapendula and other plant species that have evolved leaf Nyctinastic movement. So the question for my study is, what is the effect of varying light quality on leaf Nyctinasty movement in East Acacia or Senapendula? So for my study design, data was collected on the angle of leaf opening over time on the Senapendula species at the Eunice W. Smith Lake Field Station. The leaves were tested with changes in the sunlight's wavelengths or light quality by fully or partially obstructing and filtering out certain wavelengths using opaque or translucent colored plastics. The boxes were made from different colored translucent plastics including red, green, and blue, and clear and opaque were acting as controls. The opaque was added as a control to test whether the independent variable of varying light quality was actually acting on the angle of leaf opening and not another confounding factor. From this, changes in the angle of leaf opening and how delayed it was in each treatment was observed, investigating how the plant circadian rhythm can be altered with varying light quality. In total, there were 5 boxes covering the leaves on a Senapendula branch in a similar position and angle to ensure equal amounts of sunlight were received with each treatment. This was then repeated for each of the 3 Senapendula replicate trees, which were labeled P1, P2, and P3. Data collection procedures. Data of the angle of leaf opening was measured by taking photos of the leaves within each box treatment at 6 a.m., which is sunrise, 12 p.m., when the sun is at the highest point in the sky, 6:30, which is at sunset, and 10 p.m. at night. This was done over the span of 2 days and 2 nights. These photos were then imported in the ImageJ software, and the angle tool was used to measure the angle of the leaves for each treatment per replicate. The data was then collected and arranged in a data spreadsheet for further analysis using R. In R software, the data will be interpreted with time series graphs with leaf angle over time with each treatment as a separate line on the graph, as shown below. This is to convey variances of the adaptation between individuals in the plant species. Another time graph would be getting the averages of the angle from P1, P2, P3 for each color box treatment over time. This is to see the overall effects of varying light quality on the Senapendula species. A linear mixed effects model or repeated measures ANOVA test will also be used to account for variation among leaflet samples.